Deepak sir, I had sent you WhatsApp.
Okay, I will just.
Good morning, students. Please inform to your friends. Let them join as early as possible. Please check it. I have given YouTube Live and Facebook Live also. You can go to there and you can watch there also. Okay. Please inform to your friends within 5 minutes the section will be start because sorry for the delay because the power issue is there here. You can join fastly. Okay. Thank you, students. Good morning, students. Please inform to your friends. Let them join as early as possible. Within 2 or 3 minutes, the section will be start. Sorry for the delay because of power issue is here. You can join YouTube Live and Facebook Live also. It is going on. You can watch there also. Okay. Thank you. So sorry for the delay. Our sir is connecting now. Our ED sir, Deepak sir. Students, please inform to your friends. Let them join as early as possible. Within seconds, the section will be start. Who are willing to join in AirMeet, I'll send you the link in YouTube and Facebook. Please check it. Any students, you are having any laptop means you can join there. I'll send the link now. AirMeet link I had sent you. Please check it in Facebook and YouTube. You can join in AirMeet also.
Quick brief about our company. Epartial
Good morning, students. Today our topic is webinar on outline of drug discovery and development. Our speaker is Dr. Deepak sir.
Yes, ma'am, I'm there. Very good morning.
Yeah,
optimization, everything.
Good morning to all students. Today our webinar, we have conducted a webinar on outline of drug discovery and development from APSSDC skill development. ED sir will be speak now. Our ED sir is Ramkot Reddy garu. Sorry for net issue. Sir has disconnected again. Sir will join. He join. Good morning, students. Today our webinar is webinar on outline of drug discovery and development. Our speaker is Dr. Deepak sir. Now ED sir will talk. Ramkot Reddy sir.
Yeah. Good morning, everybody. On behalf of APSSDC, I welcome all of you. I welcome Deepak Throught, who is the key resource person for this event. Key research person for this event. The today's event is webinar for drug discovery and development, which is which will play a key role in pharma industry and its aligned industries. All of you know that the Andhra Pradesh and Telangana is the front runners in pharma industries, and 70% of pharma production will takes place only in these two states. So, as this is the drug discovery as well as development, will plays a key role in upcoming pharma industries. So, I welcome Deepak ji for giving a wonderful introduction, starting from introduction to drug discovery and development case studies, till up to the case studies he will introduce in this area. More than 1,600 students have joined in this webinar. It itself indicates that how important for pharma and chemistry students. So, APSSDC will always runs for to introducing the new courses in pharma with the directions of our Honorable Chairman, Sri Challa Masudan Reddy Garu. He himself initiated this pharma sectors to be taken up by the AP Skill Development Corporation. With his initiation, today APSSDC has MOUs with all major pharma industries, starting from Aurobindo, Lawrence, Ramki. Ramki is the sector where nearly 90 pharma industries are located. Heterodrugs, Dr. Reddy Labs, and Divi's Laboratory. We are making MOUs with almost all the pharma giants, and we have initiated industry customized skill training and placement activity where we are placing nearly 3 to 400 students per month in pharma industries. So we are also planning to introduce one pharma course, 6 months pharma course to meet the pharma regulations for non-pharma students, non-chemistry students to enter into the pharma industries. Similarly, we are introducing several varieties of the program in pharma with the initiation of once again our chairman Sallam Masudan Reddy Garu. He is particularly interested to develop a number of courses and number of programs in the pharma sector. So, as initiation of that, in that we are starting this drug discovery and development program today. We are here. I am very happy to participate in this inaugural session, and I hope all the students will be benefited and they will get the maximum knowledge from Deepak sir. Deepak sir, I request all the participants to participate daily from block your calendar from 11 to 1 p.m. regularly and listen to the research persons what they are delivering and interact them. I request Deepak ji, please give your email and still continue in contact with our participants so that they will be get benefited for a long time. So with this inaugural, with these few words, I welcome the resource persons as well as the participants who are making, who will make this program to be a useful program for the society and for the Andhra Pradesh. I will thankful for our chairman for giving this opportunity to conduct this such type of programs. Thank you very much, sir. Hmm?
Thank you, sir, for a brief introduction about Deepak, sir. Deepak, sir, you can introduce yourself and we can continue the section.
Thank you, ma'am. Let me just Maheshwari, can you just confirm if you're able to see my screen?
Yes, sir.
All right. At the outset, I would like to thank APSSDC, Andhra Pradesh State Skill Development Council, for this wonderful initiative what they have taken in form of e-learning, uh, and this skill development initiative by APSSDC, I think it is one of its kind across country, and I'm so glad that I got this opportunity to be a part of it. So let me first of all congratulate APSSDC for such an initiative, because knowledge is everything in today's time and era, and especially in this COVID pandemic crisis situation where most of the things have gone virtual. I'm so glad that APSDC has taken this initiative to conduct this skill development via e-learning for the benefit of the students across Andhra Pradesh. So congratulations to APSSDC. I would especially extend my thanks to Dora Venkata Ramakoti Reddy sir for my kind introduction and humble words you have shared. And it was really enlightening for me also to learn the efforts by Andhra Pradesh state government through APSSDC for skill development, especially in pharma sector. I must say, sir, that in current pandemic crisis situation, the only sector which is really demand for now and where the whole world is looking forward to for the solution of COVID, I think sciences sector. And I'm so glad that APSSDC did realize this, and you have taken initiative for training of people, especially in this sector. So once again, Reddy sir, thanks a lot, and congratulations for this initiative. I would also like to thank Gopinath, Mr. Gopinath, and Mr. Venkata Pratap Reddy sir for organizing this e-learning module, especially for the benefit of the students. Again, special thanks to Maheshwari for all the IT supports. And at this point in time, I would like to take this time to acknowledge and appreciate the efforts of Life Science Sector Skill Development Council representation in form of Mr. Jahangir, who is a cluster head for Andhra Pradesh. I think credit goes to him also to really work in close collaboration with APSSDC so that such skill development activities can be organized for the benefit of the students of the state. So, with that brief, I think I would like to get started with today's topic. And in next 15 days or so, drug discovery and development, I would like to cover that in brief for the benefit of the students. And for students, I would just say that, friends, even in this pandemic crisis situation because of COVID, as I was telling you earlier, the whole world is now looking forward to pharma sector for the solution of it. Be it the treatment that could be in form of any new drug development for the treatment of COVID, or the existing drug which can be repurposed. So we call it as repurposed drugs. All of you have heard about hydroxychloroquine, which is a repurposed drug which was initially used for COVID symptomatic treatment. So like that, again, it comes to the research, drug discovery, development, research only for as a solution for COVID treatment. And now, as you all of you are aware, that vaccine is the only solution, at least for now, for COVID. You will see that even that comes within the purview of pharma sector only. So I think pharmaceutical sector is a very important sector. It has always been, but in this pandemic situation, the importance is really great. So I'm glad that such a topic is picked up by APSSDC, because for students, the pharmaceutical students, graduation in pharmacy or masters or PhD scholars, or from chemistry and biotechnology students, I think this is very important for you guys to understand the role of drug discovery and development. And then once you have an understanding in this, I'm sure this will really help you in your career, or even if you want to pursue further studies. This understanding and overview of drug discovery development will help you. So with that brief, I think let's get started. This is a brief biography. I don't want to get into detail, but since I'm going to be your instructor for next 15 days, just would like to share that I'm from drug discovery development field. More than 20 years I'm into pharmaceutical research and development field, and I've worked with companies like Zydus Research Center. Zydus, for sure, all of you should know that they are one of the.
Excuse me, sir. Can you hide the button?
Yes.
Sorry for disturbing, sir.
No worries. So I started my career with Zydus. So Zydus basically is one of the company for today's scenario. You should know that from India there are three vaccine giants which are really coming up for COVID. So one of the vaccine is likely to come from Zydus. So that is Zydus. So I started my career from Zydus as a center. Then most of you are from Andhra Pradesh, so you should know. I was also a part of Dr. Reddy's laboratory, which is one of the leading pharmaceutical company from Andhra, and then of course it is for the entire country. One of the leading pharmaceutical companies, Dr. Reddy's, and then I was also a part of Biocon at Bangalore. And then I had a couple of affiliations with contract research organizations like Venta Bioscience and Origin and Microtherapeutics Research Lab in hospitals. So basically, for any drug discovery development scientist, I would say the only satisfaction we look for is if we are able to be a part of a project where a drug can be discovered and we can be a part of the entire process of drug discovery development, and finally a drug can be launched into the market. So I was fortunate to be a part of such program, and the drug is called as saraglitazar. So I was a DMPK scientist responsible for generating all the ADME and IND enabling data for saraglitazar, and it is a drug for dyslipidemia, metabolic disorder, and it is a PPR agonist drug, which is by far till today the only chemically synthesized drug manufactured, discovered, and approved from within India in 2013 by DCGI. So I was a part of that team, and with Hyderabad, I got a close connection because there's a company called Karyoline Biologicals, a biotech company from Hyderabad, and we have worked in collaboration, and we came up with India's first enzyme mixture for a veterinary application called Karyoline. So that was my little contribution towards novel discovery of drugs or enzymes. And my entire career is completely into drug discovery and development. My forte is drug metabolism, pharmacokinetics, toxicology, and efficacy pharmacology. So, with that brief background of mine, let's get started. So, as the title suggests, it's basically it's an outline of drug discovery and development. So I would call it as it's an overview of drug discovery and development. So within this purview, what in next couple of weeks of time, what we will be covering is first of all a detailed process of drug discovery and development, and then what are the different phases of drug discovery. Then there's something called a structure-based drug designing, computer-aided drug designing, and any drug discovery project starts with a target selection. So we will be talking about and learning about how a target selection happens, how actually the target gets validated, and then once it is validated, there is a whole set of in silico assays which takes place, in silico screening of molecules, followed by in vitro. So when you say in vitro, these are all the experiments which you can do in the lab scale. So those are all in vitro experiments in cell-based or enzyme-based models. And then we will also talk about in vivo studies, which is in live studies, in live animals when the studies takes place. We will also cover about drug metabolism and pharmacokinetics. We'll talk about efficacy studies, because it's very important that you should know what is the mechanism of action. And for that, the studies which we do, they are called as efficacy studies. And finally, safety and toxicology studies, which are very important, because any drug, when it comes into the market, we want that this drug should be safe and efficacious. So we'll be learning about those studies also. And then finally, after all the preclinical phase, the drugs are being tested in human. So that we call it as clinical trials. So we will be covering about in brief about clinical trials. And then today's scenario, because I always believe that we should be up to date, we should keep ourselves up to date with most recent advancement in technology. So I would also like to cover about high throughput screening, because all of you are students pursuing graduation, post graduation, or PhDs, and if you want to get into industry, then today's scenario is of high throughput fashion, where we want to do on a large scale drug discovery, screening, or development of molecules. So we want to do in high throughput fashion. So you should know about the latest platforms available for that. So we'll talk about that. And one of the most important thing is I would like to cover within this 2 weeks time, what is the role of different disciplines within the life sciences sector, say chemistry, pharmacy, biotechnology, microbiology, all these disciplines, how they actually contribute in drug discovery and development. That also we'll try to cover. So this is in nutshell about the entire course content which we'll be covering in next 2 weeks of time. Okay, and I'll try and make sure that it will be very precise, and it will be in a way that you understand the concepts, and it will be useful for you going forward, either in your career or if you want to pursue further studies. So that's about the whole course content in detail. So, especially today, being the first day of our this e program, the e-learning training program, we will first talk about the Life Science Sector Skill Development Council, because that's what I represent. And since all of you are science students, you should also know about Life Science Sector Skill Development Council, because this is uh, this is straight away related to the pharmaceutical industry of this country. I will talk about it in few slides about council. I will also talk about few slides about the organization which I represent, Prerna Bioinnovation Research Private Limited, for the purpose, because this is also a contract research organization. So just the once I talk about this organization, you will see how exactly a research organization looks like and what are the things involved in that. And then finally, towards the major portion, we will concentrate on introduction of drug discovery and development within today's lecture. Okay, so I think we are at 11:45 or so. So in next hour or so, I'll try to cover all these three topics, and then we will stop for last 5-10 minutes if there are any questions. So like this, as Reddy sir has already mentioned, we will continue this for next 15 days, and I will try my level best to make sure that your time is really utilized in the best possible way, and then the knowledge which you gain would be really useful to you going forward. So let me start with Life Sciences Sector Skill Development Council. So this whole initiative is in collaboration with AP SSDC for this e-learning program. So Life Science Sector Skill Development Council is basically a non-profit society. It is actually a national level certification body under the mandate of Ministry of Skill Development and Entrepreneurship, and this comes under NSDC, National Skill Development Corporation, and it is promoted by Confederation of Indian Industries. Okay, and basically within the governing body of Life Sciences Sector Skill Development Council, we have representations from almost all the pharmaceutical associations of our country, like I will come to the detail stakeholders detail, but just in nutshell, we have the representation from government as well as the major industries. Okay, so as a life science student, you should know that India is the third largest exporter of vaccines. In fact, I would go a step ahead and I'll tell you that it is one of the leading biopharmaceutical sector in our country, in the world, I guess, because more than 60% of the world's requirement of the vaccine is fulfilled by India. Okay, and I would say that even for World Health Organization and UN, their vaccine demand, more than 50% of that demand is met by Indian industries, be it Bharat Biotech, Serum Institute, Indian Immunologicals, but all these vaccine manufacturers, they belong to India, and more than 50% of World Health Organization's vaccine requirement is met only by Indian companies. So that is our contribution in life sciences for the world, right? So what is the aim of life science sector, LSSDC? So our only sole aim is to actually fill the gap between academia and the industry requirements. Because friends, what happens is, although we study in our college and universities as per the approved courses, but for some reason, many a time it happens that the requirement of the industry is slightly different than what we are studying in the college. Of course, whatever we study in the college is of utmost importance, and it really helps in understanding, in improving the understanding of the whole pharmaceutical principles. But sometimes they're not aligned to the industry requirement. So that is the job of Life Science Sector Skill Council. We try to identify what is the requirement of the industry, and then we work closely with academia so that we can have industry ready candidates. So at least when students are graduating or doing post graduation, they acquire those skills, they learn what is required in the industry, and then the placement and everything becomes easy for the candidates, that is you guys students. So that is the job of Life Science Sector Skill Development Council. And what we have done is, like, we did a survey pan India as a council, as a government initiative. And what we found was there are around 100 job roles which are prevailing in entire industry, pharmaceutical industry. So out of this 100 job roles, at least 60 of them are comprising more than 95% of the job roles demand in the industry. So what we did is we enlisted those 60 job roles, and then we prepared the QP, which is qualification pack, the course curriculum for those job roles. I'll talk about those job roles in a while, but we could develop the course curriculum for those job roles. And now those job roles are available for academia. It's on the website of our Life Sciences Sector Skill Development Council, and they can pick up those job roles, and they can use those job roles to train their student as and when required. And I'll tell you the importance of council is, most recently, just in one last month, there has been a development that Pharmacy Council of India has now given a circular to all the pharmacy colleges pan India, that in their 8th semester of B Pharm course, generally 2 elective subjects are taken. So one of the elective subject now they can take from Life Science Sector Skill Development Council courses. So here onwards, I won't be surprised if going forward it will become mandatory that most of the pharmacy college will have to take one LSSDC course in their 8th semester as an elective subject. So that's how we hope that students will have the required training as per the requirement from the industry also. Already they have passed on an advisory that within the pharmaceutical industry, all the manpower, the preference should be given to LSSDC certified manpower. So that is what we are working with CDSCO also. Up till date, while I'm talking, again, this is a little old slide, almost 2 months ago. We have more than 50,000 pharma employees already certified by Life Science Sector Skill Development Council. On this slide, if you see, with the council, we have more than 300 training partners, more than 640 industry partners. We have, as I was telling you, more than 60 qualification packs already. RPL is the existing manpower in the pharmaceutical industry, which is called recognition of prior learning. More than 50,000, as I said, we already been certified. We have more than 11,000, which is again an old figure. We have now close to 15,000 apprentices from life sciences sector only. So, friends, let me tell you, this concept of apprenticeship was, of course, it is by law, every company should have apprentices. But in life sciences sector, the companies used to keep it to as minimal as possible, that is 2.5% of their total manpower. But now, with the involvement of council, the companies, be it Dr. Reddy's, be it Mylan Labs, be it Zydus, Cipla, Sun Pharma, Torrent, all these companies have now started taking up to 10% or 15% of their manpower as apprentices, all from life sciences sector only. Means you guys from pharmaceutical area, they have scope now to get into companies as apprentices. More than 20,000 placements we have achieved so far through the council, and we have more than close to 400 trainers who are certified by council up till date. So when I say life sciences, within life sciences, it includes pharmaceutical, biopharmaceutical, and contract research organization, which includes API manufacturers, nutraceutical companies, biotech companies, biopharmaceutical companies, discovery, as well as clinical research organizations. All of this comes under the purview of Life Science Sector Skill Development Council. Just to name few major companies like Sun Pharma, Lupin, Dr. Reddy's, all these are the major players from our country. And that's how India is the third largest pharmaceutical industry in the world. And for all of you students, you must be already aware. But within India, if I talk of clusters, I think Andhra Pradesh, from the southern Andhra Pradesh, contributes to be one of the very major cluster for pharmaceutical companies. You have best of the companies based out of Andhra Pradesh, especially in Hyderabad. You have Dr. Reddy's, Milan Labs, Hitro Labs. Uh, you have many of the CROs operating out of like Suin Labs. Like there are many pharmaceutical companies operating out of Hyderabad and nearby areas. So even Andhra Pradesh accounts for one of the important clusters in terms of pharmaceutical industry. So in as I said, it's an initiative of the government. So we work very closely with different government departments like CDSCO, Central Drug Standard Control Organization, also with Ministry of Chemicals, with Pharmaceutical Department of Pharmaceuticals, and DBT, Department of Biotechnology, and within the governing board of council, as I was telling you, we have representation from all the major associations from pharmaceutical industry like Bulk Drug Manufacturer Association, Federation of Pharma Entrepreneurs, Indian Drug Manufacturer Association, Indian Pharmaceutical Alliance, Organization of Pharmaceutical Producers of India, Association of Biotechnology-led enterprises, Pharmaxil, and then I must tell because I'm talking on behalf of APSSDC that LSSDC is headed by the chairman, Dr. Satish Reddy. He is the chairman and managing director of Dr. Reddy's lab. So he himself is the chairperson of the council. So on this slide, I will like to spend few seconds, like 15 to 20 seconds, for the benefit of the students. This will actually tell you the requirement in the industry. So by far, if you see, more than 48% requirement in the industry. I'm referring to only life sciences sector. Okay, within the life sciences sector, in industry requirement is 48%, like almost 50% is in the manufacturing field. Okay, when you talk of research and development, around 10% requirements are there in the research field, around 24% is again in the marketing field, and of course quality assurance or that field is also equally important, around 5% requirements, and like supply chain and other things constitute for 4%. So in nutshell, more than 50% requirement in industry is only in manufacturing area. If I go by qualification, because all of you are students at and pursuing your education at different levels, you should know the distribution of manpower within life sciences sector industry. So around 5 to 8% of the manpower pan India across life sciences sector is PhDs or postgraduates. Okay, for the graduations, they constitute around 15 to 25%. For ITIs, for DPharm, for diploma, around 15 to 20%, and for graduates, again around 25%. So this is the whole distribution based on the qualification within the industry. Okay, so I hope this will help you to identify where exactly you fall. But in nutshell, the take-home message is that doesn't matter if you're doing graduation or even if you're doing ITI or a diploma or engineering or post graduation or PhD, as long as you are into life sciences sector, your life is secured in terms of your career, and I'm sure all of you will have a great scope to get absorbed in industry if you want to get into industry, and there is equal scope in academia also if you want to get into academia. So, coming to a few of the programs I was just talking about in life sciences sector, we are running this apprenticeship program where when you are pursuing your final year studies like graduation final year or the post graduation final year, uh, 6 months of industry training is required, right? Or even after that, if you really want to get absorbed into industry, then you can enroll yourself as apprentice, and you can be a part of any industry within the local area you can prefer, and then you will be taken as apprentices. You'll get some stipend from the company, and after successful completion of apprenticeship period, you can be absorbed as employee, as a full-time employee on their payroll. What are the job roles? As I talked about, 60 job roles identified pan-India across life sciences sector. These are just to name the few, like bioprocess engineer, manufacturing assistant, quality assurance chemist, scientific medical writing, production biologist, QC biologist, QC chemist, store assistant, manufacturing assistant, packaging assistant, maintenance assistant, medical sales representative. So, like this, there are 60 job roles which can be seen on the website also. Licenses sector is also running e-certification program, which is an online program for pharma production, medical sales representative, and quality analytics. We are also running one program, just like APSSDC has taken this initiative, called Epartial, and then where we do an accelerated 1 week programs on drug discovery development, analytical technologies in drug discovery and technology. Licenses sector is having 5 center of excellence across country. Two of them are in Gujarat. One is like Kedila Healthcare Limited, and we have Macleod Center of Excellence. We also have in Karnataka Micro Labs, nominated as Center of Excellence, and in Telangana we have Swarna Bharat Trust, initiated by Granules India. But Granules India is also one of the very important company doing a lot of skill development activities, and we have Macleods in Himachal. So these are the center of excellence for life science sector. And this is about ePathshala, 1 week accelerated programs, drug discovery and development, analytical technologies. Okay. So with that brief about LSSDC, let me very quickly take you through Prerna Bioinnovation Research Private Limited also. And there is a purpose because I just wanted to, just through presentation, I wanted to showcase to you typically how any research company looks like. Okay, and then we'll jump into our topic for today, that is drug discovery and development introduction. Okay, so Prerna Bioinnovation PBR Private Limited, to which my affiliation is based out of Gujarat, Gandhinagar. It is a preclinical contract research organization, and we provide solutions in analytical, bioanalytical, microbiology, biotechnology, pre-formulation, DMPK, pharmacology, and toxicology research and training. And we cater requirements, testing requirements from pharmaceutical, medical device, cosmetics, biotech, agro foods, feed supplement, herbal, and chemical industries. This is just a pictorial presentation, how a lab looks like, a research lab. In your college also you may be having a labs. So this is typically how a research lab looks like. There is also a CPCSA approved animal facility. So this is how an animal facility looks like, with epoxy floorings, and there are guidelines of CPCSA under Ministry of Environment and Forest. So it is a state-of-art facility as per the mandate from the CPCSA. So these are a few pictures from the where actually the testing, the live testing on animal actually goes on. Okay. One of, as I was just talking about, one of the very important project, a research project which we got success was Karyoline, which is India's first enzyme mixture, which was launched in 2016 in collaboration with a biotech company from Hyderabad called Karyotica Biologicals. This is called Karyoline. So, friends, as I was telling you that for research, you need to identify a clinical need. So, like in milking animals, milk production is one of the important factor, right? Milk production with good fat percentage. So, what we found through research and through literature review, that the digestion is a major concern in milking animals. Like more than 50% of the food which they eat, the feed they eat, is going unabsorbed in their feces. So whatever efficiency in the animal we are getting in terms of milk production or a fat percentage is all because of only 50% of digested feed. So that's a clinical need we identified. So we just thought that if somehow we can increase the digestion of the feed stuff, maybe their efficiency can increase. So that's how we came up with this solution in form of 9 enzymes cocktail, like cellulose, hemicellulose, xylanase, protease, lipase, pectinase, amylase. we just thought that if we put this all enzymes together, and why we selected these enzymes is because across country, whatever different feed stuff is served to animals, majority, more than 95% of them is digested by one or the other these enzymes mentioned as per here. So we thought if we mix up all these enzymes, then that formulation would be applicable pan India. So that's how this Karyoline was developed, and you'll be surprised that but just by giving this, the digestion increased almost by 25%. So earlier 50% digestion efficiency was there, now it has become 75%. So with that improvement in digestion, there was increase in milk yield, there was increase in fat percentage. So that's the concept of these enzymes. This is the function when we actually launched it officially on a government function, and it was duly covered in newspaper as well. The only reason is because, like as you know, there are malpractices of giving hormonal injections to animal to increase milk production, whereas this is purely a natural mixture of enzymes. Just by improving the digestion, you can increase the efficiency of animal. So it was duly covered in the media as well. Most recently, in this environment of COVID, we came up with this. Again, as I said, this is not a novel discovery. Like UV lights, we have been using for many years. I mean, all of you who have worked in microbiology or in pharmacy college, you will know that you have laminar airflow, right? So we use a UV light. So what is the purpose of UV light? It makes your area sterile. So it will be free of bacteria, free of viruses. So what we did is we just thought if we can make this and we can use UVC light. So UVC light is nothing but the emission at 254 nanometer. So what happens is at this nanometer, this UVC light is germicidal, and it is proven. It is already literature proven, and it will kill the bacteria, virus, or any live cell which comes in contact at UVC frequency of 254 nanometer. And the purpose here is now anything which is like your keys, your wallet, your currency notes, anything mobile, whatever is exposed to external environment, you just put it inside, switch on the UV, and within 8 minutes it will make it completely sterile. Any virus, including coronavirus or any other bacteria, would be killed. And you just have to be careful about one thing, that it is hazardous, UVC light, all of you know, because it cannot differentiate between a bacterial viral cell or a human cell. So it will simply kill it. It will simply destroy the RNA, DNA inside that. So the only reason is you should be cautious. Never put your hand inside while the light is on. So while the instrument is switched off, you keep your stuff inside. Switch on the instrument. After 8 minutes, switch off the instrument and open and take out your items or articles, and everything will be sterile. I must say that since last 6 months, personally, I'm using this, and I'm using very little of sanitizer or liquid hand sanitizers, especially for keys, wallet, currency notes, watch, mobile, eyeglasses. Everything is getting sterile in this. So we just wanted to make a cost-effective solution, and so that's about. Again, this is one novel thing, so I thought it would be important to share. And industrial training is one of the very important thing which we impart from here, because as you know, most of you have to do your industrial training dissertation work in your final year. So we have done many dissertation M.Pharm research work. Of course, we don't do it; we make the student do it. But since our experience, with our experience and expertise, we guide them to select a novel problem which can be useful for the society, or at least it is from a research perspective, it is novel, and we help them write a thesis out of it, and we also help them in writing the paper out of that. So we do the summer trainings, also industrial training very often, where we take them through theory lectures, but with practical exposures as well. So these are just few snapshots from all the trainings which we have done as recent as just a couple of months ago in COVID environment. We did it for a limited number of students with social distancing and all, but we have completed this study. So we help B.Pharm students, M.Pharm, and PhD students in pursuing their research or dissertation work also. So with that brief introduction, I think let me jump into our today's topic, which is drug discovery and development. I'm sure since all of you are pharmacy or science students, you might have heard about these terms, right? Drug discovery, ADME, which stands for absorption, distribution, metabolism, and excretion. In vitro assays, in vivo assays, PKPD, toxicology, safety studies. So I'm sure in one or the other subject during your studies, you might have covered this. But what we will try to cover is from a practical perspective, we'll try to cover these topics. Of course, we'll talk about the basics of that, but we'll see what is the application of this. You might have studied what are these assays, what are these studies, but you should know how they're applied in industry, because that's where your career starts. When you get into industry, you will be expected to utilize these assays for industry projects. So I'll try to make it as practical as possible so that it can give you insight into the particular topic in detail. Okay. So, what is drug discovery and development? So, friends, for me, drug discovery and development is like a roller coaster ride, right? Like all of you guys, I'm sure, would have at least tried this roller coaster ride once in your lifetime, right? We just love that. We completely enjoy this roller coaster ride. Why? Because it is full of uncertainty. You never know. Sometimes it's like it's all of a sudden it will go down. Sometimes it goes up. Sometimes it is upside down. So you are screaming out of joy, right? So that uncertainty, you don't know when it's going to swing around, when it's going to go down. That really makes roller coaster ride interesting, and that's the same thing I call drug discovery and development as. Okay, because there are so many fields, multiple disciplines actually are involved within this. Okay, and then we are really taking this as an interesting thing because it is highly uncertain. Whenever you start any project, you never know whether you will succeed or not because there are so many unknowns, and that really makes this entire field very interesting. I just put a few areas name, like any project in drug discovery and development starts with identifying a clinical need. Means you should have a clinical need, like what is the disease you are looking for. Is it COVID? Is it diabetes? Is it cancer? Is it inflammation? Is it autoimmune disease? So you have to identify a disease. Then only there is a clinical need. Only then your project can be started. You should also look if it is a novel project, if it is a novel drug which you are trying to develop, is there a possibility to do patent? Because if you don't look into that and you waste your time, energy, resources into the project, and then going forward, if you come to know that somebody else is already having a patent, then all your efforts will go waste. So you should also look for whether there is a patentability or not. If you have an entire preclinical research plan in place, don't worry, we will understand what do you mean by preclinical plan. You should know the regulatory pathways, because if you want to register your drug in India, then you have to go by regulatory guidelines run by DCGI. If you want to register your compound or a drug in US, then you should have an understanding about requirement of regulatory requirements from FDA. If in European country, then it is EMA guideline. So based on your filing or where you want to register your product, you should have an understanding and a plan for regulatory pathways. A business strategy, of course. Without business strategy in place, no project can succeed. And most important backbone for any project is finance. So you should have a financial plan in place. The entire designing should be there for the product. Finally, you should have a plan to conduct a clinical trial. And after doing all this thing, when you actually launch your molecule or a drug in market, you want to look for reimbursement of your investment. So you see so many different things, and your project can fail at any of these phases. And that's why we call it as roller coaster ride. I think somebody has just asked a question about PharmD. So I would mention it here that yes, this is very much relevant to for a PharmD student as well. Okay, this is relevant to any pharmacy student, any license sector student, which includes pharmacy, PharmD, D-Pharm, B-Pharm, M-Pharm, PhDs, or a science student in chemistry, biology, zoology, biotechnology. This is completely applicable to any of you guys. So on this slide, this is just a cartoon. As you can see, doctor is getting ready with his syringe, and this small kid is asking his mother that I'm going to get this injection in my buttock, but I'm having a sore throat. So how that drug will travel from here and will reach my throat? You see, so that is actually because he's a kid, he doesn't understand. But this is just what we are trying to understand through DMPK. So in this couple of weeks of time, my whole purpose would be to make you understand how exactly this whole thing happens, right? So even since the currently COVID period is going on, so in one of the lectures, I would like to cover about the vaccines also. And as all of you are science students, I think all of you should understand about vaccines. And especially in COVID environment, I would like to cover the different vaccines from different companies, from Indian companies, including Serum, Bharat Biotech, or Zydus Research Center, or from Pfizer, or from Moderna Therapeutics. We'll try and understand what is the difference between these vaccines and how they are useful. Okay, so as science student, I hope all of you should understand that. Okay, so this is another cartoon where you see the scientist has put up this blindfold, and he's just adding drugs and then waiting for discovery by chance. Of course, I've taken it as a cartoon, but it's a reality. All of you know how penicillin was discovered, right? Penicillin was never discovered. It was never the intended drug for which scientists was working. It was a contamination found in a plate, and somehow later on, the scientists realized that, oh, this is a penicillin and it can act against against bacteria. So that was a discovery by chance. But you cannot actually organize this in your lab and try and do discovery by chance. Okay, so that's about discovery by chance. And finally, this is again a cartoon, but very very true. I guess we should know, all of us should know about it. The patient is actually lying on the bed, and to the doctor or the nurse, he's saying that I stopped taking medicine because I prefer original disease to the side effects. Very true, right? Many a times it is true. Although we work in drug discovery development, but this is a reality. Many a times our drugs which we take, they have a lot of side effects. So the patient is saying that I stopped taking drug because disease is better than the side effects of the drug. So, but yes, as a scientist, I would like you should understand, if at all there are any side effects, why those side effects comes, how do we minimize those side effects, and we should understand the disease progression also. So this is just a cartoon, but yes, we will understand the science behind that. Okay. So what is drug discovery and development process? So you can imagine this and as a process which can be divided into different phases. Okay, it can be as a discovery phase. You can have an early preclinical phase, a clinical phase, and then finally an approval phase. So the whole process is very lengthy. Okay, it takes around 10 to 15 years to do the drug discovery development, even for one novel molecule. Please understand, to bring one novel drug successful candidate into market, it takes around 10 to 15 years, which is a long period. You see how much investment it takes. It takes around $2 billion of investment. You see, it is a very costly affair, and that's why drugs are costlier, because in order to bring in one novel drug into market, you should work for at least 12 to 15 years, right? And what is the success rate? It is 0.003%. You see, so less a success rate. So the success rate of less than 0.5% is what is there in drug discovery and development. That is, when you start working on 30,000 molecules initially, there is a chance that you will have one successful molecule which can be launched in the market. That's why 1 by 30,000 is 0.003%. So regulatory agencies and pharmaceutical companies, they all want safe and efficacious drug only to come into the market. But it is very easy to say But to actually practice it and get a safer and efficacious drug in market, this is the success rate you have, right? And of course, there are reasons why drug fails. Why this out of 30,000, only one drug will come up and rest 29,999 drug will fail. There are reasons, we'll understand that. But for your understanding, you should know that drug discovery development, it takes 15 years of time and more than $2 billion of investment. Just try to explain on this slide little bit in more detail how this success rate is less. So you see, out of 30,000 molecules a chemist will start synthesizing early in discovery, only 2,000 will enter into preclinical development. You see, preclinical means this phase where you can do in vitro and in vivo testing in animals. Only 2,000 molecules enter in that. Out of that, you get into human trials, phase 1, only 200 will get into phase 1 clinical trial. Only 40 of them will get approval for phase 2 trials. Only 12 of them can get into phase 3. Out of that, hardly 8 molecules will get approved, and out of that, the drug where you get return on investment would be hardly 1. So that's how this success rate is too low of 0.003%. Let's talk about different phases. We have talked about different phases of drug discovery and development. Let's see what they are. So, in discovery phase, there is early drug discovery where you do a target identification and validation. Okay. So, as I was telling you that you should know, you should have one disease. You should identify a disease, a clinical need, a target on which your drug is going to act. So that is called target identification. Say for diabetes, we are taking 10 different types of drug. Each drug can have different mechanism of action. They can have different molecular targets. Eventual outcome would be reduction in glucose, but it can act on different targets. So that is called target identification. It's not only identifying, you need to validate that target. Then you have to do high throughput screening or a hit discovery, and you develop particular in vitro assay to test the drugs and do the screening of a molecule. As I was telling you, if you do it in high throughput screening, then many compounds can be screened, and then finally you come with one lead compound. Okay, and that lead we try for optimization of that lead molecule. So you see, in early drug discovery, target identification, hit discovery, assay development, high throughput screening, hit to lead stage, and lead optimization. Then we get into preclinical testing. So preclinical, as the name suggests, preclinical means before clinical studies. What all studies we do, they are called as preclinical studies. So which includes in vivo studies. In vivo means in live studies in animals, which could be laboratory animals, which could be rats, mice, guinea pig, hamster, rabbits, beagle dog. All these studies are done, they are called in vivo studies. We also do many in vitro studies. What are in vitro studies? This could be cell based studies, this could be enzyme based studies. So we will be doing in vitro studies. Then we do ADME studies. What is ADME? Absorption, distribution, metabolism, and excretion. In nutshell, if I had to give you an example, if you take a paracetamol tablet, suppose you're having a fever, you take paracetamol tablet, right? What do? We take it by oral route. It goes to your esophagus, goes to stomach. From there, it will reach your intestine. Within intestine, it gets absorbed by intestinal barrier. That is called absorption. After it gets absorbed, there are vasculature through portal vein. From intestine, it goes to your liver. Okay. In liver, it will get metabolized. So that is called metabolism. After metabolism, it will get distributed to the rest of the body. That is called distribution. And finally, through kidney, either metabolite or the drug itself can get excreted out of your body. That is called excretion. So we do absorption, distribution, metabolism, excretion studies. Then you will do a proof of concept study. Say for example, I come up with a concept that okay, my drug is having anti-diabetic activity. So it is able to bring down the glucose level. Then I have to prove this. I have to have a disease model, at least in vivo model in animals. Only then I can do a proof of concept. Once I prove that yes, in a disease condition, I am able to cure them, or I am able to treat them, I am able to bring down the glucose in animals also in a disease model, then that will be accepted as proof of concept study. We also do a lot of experiments on drug delivery. Yeah, because drug delivery is important. You may have great drug with you, but if it is not delivered properly, say for example in today's scenario we are talking of vaccines, so a delivery route is required, right? Of course they may use a nanoparticle technology to deliver a drug, but eventually either injection through intramuscular or through intravenous or by oral, it has to be delivered to your body, right? So that delivery has to be worked out. So that also happens in preclinical. So we work on formulation optimization, then we do a dose ranging studies. Finally, IND studies are done, which are investigational new drug enabling studies. So suppose I want to do a clinical trial. So before that, I have to submit IND application. So that is called IND application. Okay. All this data, whichever I have talked about, would be submitted as IND submission data. The regulatory agency will review that, and then they give me permission to do the clinical development. So clinical development, what all things we do? Majorly, we do clinical trials. So clinical trials, you can divide it for easy understanding. There are 3 clinical trial phases, or say 4. One is phase 1 clinical trial. Please remember, it always happens in healthy volunteers. Okay, so it never happens in disease patient. It happens in healthy volunteers and a small population, hardly you say 10, 20 or less number of volunteers. When you say phase 2 and phase 3, it typically happens in patients. So in hospitals you have to recruit patients for these studies. Phase 2 again around 100 to 500, and anything above 500 to thousands number, that is phase 3 clinical trial. So this is phase 3, phase 1 and phase 2. Phase 4, I'll talk about it later. But for your understanding, once the drug is approved in the market to be launched in the market, after that we do the post-marketing surveillance. That is there any side effects? Are there any side effects of the given drug? We study those things, adverse effects, and that is called phase 4 clinical trial. So these are all the trials which happens in clinical development. We try to study and address safety and efficacy of your molecule. Then a portion is once you do this, you submit an ANDA application to regulatory agency. They will review your application, and either they reject or they question or they approve your drug. And now your drug is registered, and then you can actually launch it to the market. And phase 4 clinical trial will happen, which is post market surveillance. All right. So, in nutshell, drug discovery development can be divided into different phases: early drug discovery, preclinical. This is a discovery phase. Then you have a clinical development, and then clinical approval and post-marketing surveillance. So, just to summarize what all we have talked in last few slides, we start with identifying a target, a disease. Okay, within the disease, your molecular target, which could be an enzyme, cell, or ion channel, a molecular target. Then we validate that target. After validation of the target, we do the hit discovery. Many molecules will try to screen on that. From that, we will get our lead molecule. That lead molecule we optimize for its ADME properties, and then we take them into preclinical development in animal testing. After animal testing, all the data is submitted for IND submission. From IND submission, if it is approved, we get permission to do the clinical trials in human. After doing phase 1, 2, 3, we submit the data to regulatory agency, in this case FDA. They will review the data, and if it is approved as a drug, then it can be used to cure the disease. So you see, this is the whole cycle: target identification, validation, hits, lead optimization, preclinical development, IND submission, clinical development, ANDA submission, and approval. Within hit and lead optimization, drug discovery approach. There is something called as lead finding, new versus old techniques. We will cover that in our subsequent lectures also. These are different ways like computer-aided drug designing, rational drug designing. We will cover that thing. Okay? So, in terms of different fields, okay, all of you are from either from pharmacy or chemistry or biotechnology or science, biology, zoology background. Let me tell you where different fields are actually involved in drug discovery and development. So, when I say target selection, there is major role of genomics, proteomics, bioinformatics, cellular and genetic targets because it's going to be either enzyme, your target is going to be a molecular target, right? It can be a gene, it can be a particular ion channel, it can be a particular enzyme or a cell base, some pathway inhibition or induction we are trying to do. So here is a role of biotechnology. You see, even a microbiologist can contribute here, or a pharmacy people can contribute here. Then there is a phase called lead discovery where we do actual synthesis of your molecule, drug molecule. So that is done by chemist. So chemistry comes into the role here, either a graduate or a postgraduate in chemistry, even combinatorial chemistry. So organic chemistry, inorganic chemistry, all that comes into the play here. Again, pharmacy people can do high throughput screening. Analytical chemistry also comes into play here. As I was telling you, further also when we do structure activity studies, we'll talk about it later. We do further chemical synthesis, all that thing, chemistry comes into the play, the medicinal chemistry part. Then either a pharmacy or a microbiology people, they can do cellular assays, right? And they can do all the screening. So that is called in vitro screening. Okay. Then zoology or pharmacology or veterinary, all this field comes into the play when we do in vivo studies because they're disease models we are trying to develop. We are trying to study behavioral studies, functional imaging, PK studies. So all that thing where the pharmacology comes into the play. Finally, students who have studied clinical CRA, clinical research associate, or clinical research they are studying, they can be part in clinical trials. So you see, microbiology, biotechnology, pharmacy, pharmacology, and one important thing in all these phases, there is a quality control involved, quality assurance. So quality people work there. So if you're doing your M-Pharm in quality assurance, then you have a role to play too. So in nutshell, microbiology, biotechnology, pharmacy, pharmacology, toxicology, zoology, chemistry, all these people, veterinarians, together they can contribute towards different phases of drug discovery and development. Biologics, specifically I've kept this slide for biotechnology because nowadays a lot of students are interested in biotechnology. So you should know that even biologics, so today is a scenario of biologics. Okay, 10 years ago, most of the drugs which were developed, they were small molecules only, like paracetamol, brufen, ketoprofen, any antibiotic, cephalosporin, or metformin, anti-diabetic drug, all the drugs which are majorly available in market, they are small molecules. But now in last decade or so, a lot of development have happened in the field of biologics, biotherapeutics. So biologics, they are more complex in structure and function than the small molecules, which is the chemical drug. As you see, acetyl salicylic acid is such a small molecule. Hardly 0.18 kilodalton is the molecular weight, whereas biologics 150 kilodaltons. So you see, huge molecules biologics are. They are more complex, of course. So anything you talk in biology, say monoclonal antibodies, recombinant proteins, or cytokines, or biological immunomodulators, radiolabeled biologics, or enzymes, all these things are biologics. Okay. Even post-translational modification happens in the structure, so they are really complex. Whereas small molecules are very simple, chemically synthesized molecule. They are very simple. Okay. So biologics are made by living cells through well-controlled process. So as you can see here, the process itself starts from transfection of DNA into a host cell. You will have to select the cell line, right? Further, you actually want to have a cell bank where you want to maintain the cell lines. Then you do further fermentation. Then you isolate and purify them. You want to do the characterization, do the stability studies. Finally, it gets filled out in a formulation form. Again, you need a cold chain for refrigeration storage, and then finally it's given to the patient. So this is the cycle of a biologics, how they are made. The major difference is from a living cell, unlike a chemically synthesized molecule, which is not happening in a living cell. Okay, whereas for biologics, it is always starts from a living cell. So they are more complex too. So as a student, for you, you should understand the difference between chemical medicines. Okay, so chemical medicines, they are made by chemists, but they are small molecule, chemically synthesized. And once you have your reactions identified, then it will give you exactly same yield every single time, right? Once you have your SOP protocol in place, you can practically synthesize it exactly same yield every single time. It is straightforward process. Whereas that may not be the case with the biologics, the large molecules, because here you are trying to do something with the genetic blueprint itself, right? So any change in this entire process, in any of the steps, any change happens, the yield will come down. So there will be some variability. So that is the difference between a biologics, a drug of biologics origin, or a chemically synthesized molecule. Okay. Again, IND submission is same for a small molecule or for a large molecule. Especially for large molecule, they are interested in knowing the pharmacodynamics, PK, toxicology. Don't worry, today is the first lecture, but I assure you that by the end of this 2 week of course, all of you would know about PD, PK, toxicology, all these terms you will be knowing completely. Okay, but just trying to give you an overview that be it biologics or be it a small molecule, they all have to go through the same process of drug discovery and development. So to coming back to where we started, please remember there's something called as drug discovery development funnel. Okay, so this is a drug discovery development funnel, which is nothing but, as I was telling you, you start with thousands of molecules, and by the time you come to the end of having one molecule which is approved in market, you see all the screening have happened. You started with 10,000 molecule in discovery, hardly 250 goes in preclinical, hardly 5 gets into clinical trials, either phase 1, 2, 3, and hardly one molecule will get the FDA approval for launch. Total time, if you total it up, comes to around 15 years, more than 2 billion of investment. So this is called drug discovery development funnel. Okay, phase 1, if you do the trial in healthy volunteers, test your drug, 20 to 100 healthy volunteers is phase 1, 100 to 500 volunteers which are patients, that is phase 2, and when you test your drug in 1000 to 5000 volunteers or more, 5000 patients, then it is called phase 3. Okay, as I was telling you, this is a costly affair. Like this is an old slide, but from 1995 till 2014, actually you see from $15 billion, the investment has gone to almost $50 billion. You see, huge rise in the investment. But unfortunately, the success rate, if you can see my arrow, success rate for some reason is still plateau. It is not increasing in the same fashion. But on a not on a lighter note, I would say that all of you and us, we all should be actually happy about these figures, because you see this much investment year by year in R&D is increasing. So if you are a life science student, okay, if you are a science student, then you have huge scope. Your career is very bright, because there is huge investment year by year coming up into R&Ds, manufacturing and all. So all of your job is secured. So for us it is good, but unfortunately the success rate, that is success of developing new drug, is not same increasing fashion as the cost is increasing. Okay, so now I hope that after this much discussion, all of you should know that it's a costlier affair. Many people complain that these pills, these drugs are really costly. Okay, So why you should know now? Because to do the preclinical trials, preclinical studies, more than $300 million of investment is going on. Okay, more than $400 million investment goes into clinical trials. More than $100 million even after approval of the drug. So that's why this much investment gets into getting one successful drug into market, and that's why we see these tablets or pills, sometimes they are costlier. For one strip, sometimes it costs you 500 rupees or 200 rupees for 10 tablets, right? And many of them generic drugs, if you talk, they are cheaper. So in general, new drugs are always costlier because a lot of cost investment is there. Okay, now in next few slides, before I stop for today's discussion, in few slides, let me I have on this slide I have just talked about investment, but you must be thinking that at the end of the day you're having only one drug successful in the market, right? Then why so much of cost is there? This is the reason. You see, the investment is on 30,000 molecules. As I was talking about, success rate of 0.003%. Of course, one drug is coming in market, but you start with 30,000 drugs, and at different phases of drug discovery, they fail. So your investment goes into the drugs which fail also. That's why your cost is higher when you come up with a final product. You got my point? So that's the reason this is a costly affair. The reason is the cost of developing the ones that fail, that also adds up, right? So that's why we always say as a scientist that if drug has to fail, let the drug fail early. Not that we want that drug should fail, but if at all, for whatever reason, if it is not safe, it is less efficacious, or it is having some formulation issue or stability issues, if for whatever reason drug has to fail, I would want we all should make sure that drug fails early. So if it fails early, it will cost you less. If it fails later, it will cost you more. So a drug at a chemist shop, I mean to say, when a chemist is synthesizing, if it fails at that stage, it will cost you less. If it comes to a biologist for doing studies in animals, if after that drug fails, it will cost you more. Suppose a clinical trial is happening, after that drug fails, then it will cost you even more. So in nutshell, fail early, fail cheap. So you may read this somewhere slogan in papers and all that, fail early, fail cheap. For that reason, if your drug has to fail, make sure that it fails early so that it doesn't cost you much. Let's see what are the reasons. There are so many reasons for which a drug can fail. These are major reasons. Of course, it's an old slide. So this 41%, there was a time almost a decade ago when drug used to fail. 40% of the drug used to fail because of poor pharmacokinetic profile. We will understand what is pharmacokinetic profile later on, don't worry. But because of absorption, distribution, metabolism, excretion issues, drug used to fail. But in last decade or so, because of improvement in our understanding, because of advancement in technologies, now this 40% failure because of PK has come down to only 7 to 10%, just for your understanding. More than 22% of the drug fails because of toxicity. They may be great drugs, right? They may be highly efficacious, but they'll be toxic too. There'll be some side effects. So because of that also drug can fail. More than 30% drug fails because of lack of efficacy. Very important. Many times drug are showing great activity while you are doing cell-based assays. Okay, but when you actually do the clinical trial, it will fail because in human same efficacy is not seen what was seen in the cell-based assays in vitro or animal studies. So lack of efficacy is also one of the major reasons. Because of toxicity, efficacy, or poor PK, a drug can fail. This is just a survey which was done, as you can understand, for the drug under different phases. If they fail, what are the reasons? So, when the drug trials are going on in preclinical phase, more than 44% will fail because of poor toxicology, means because of side effects. If a drug fails in phase 1, then 27% drug or 28% drug because of safety reasons. In phase 2 and phase 3, more than 50% of the drug fails because of lack of efficacy. Okay, because here you are checking the drug in patient population, so they are our real disease target population. In that, if drug fails, then you cannot take the drug further. Finally, drug can also fail because of registration. In there also, the reason is clinical safety. Why? Because when you do a post-marketing surveillance, you may find that there are so many side effects. So drug has to be withdrawn from the market. So that could be because of clinical safety. So major reason for drug failure are lack of efficacy, lack of safety, or toxicology in preclinical phase. So to summarize, you start with I've taken the example from Pfizer. That Pfizer, they are doing many drug programs. Okay, out of that, different, say, hundreds of discovery approaches. So millions of compound they will screen. Not millions of compound will be synthesized, but they can do in silico libraries. They can screen. So millions of compound they are screening. Few of them will reach preclinical stage. Only few will reach the preclinical safety stage, then clinical pharmacology and safety, and then finally one or two product would be developed. So in nutshell, 10 to 15 years of period, more than 2 billions of investment, and screening at different phases will lead to successful launch of one or two products only in the market. This slide I have kept for purpose. This is called drug withdrawal from the market. So for a reason I have kept this slide, not to discourage you people, but as a science student you should know we have been talking about, and next 2 weeks we will talk in detail about drug discovery and development. But after doing so much of drug discovery development research, okay, after 12 to 15 years of research, be it discovery, early discovery, preclinical or clinical, when actually drug is launched into the market, unfortunately, after that also, drug has to be withdrawn from the market. So you see, this is a list of drugs which are withdrawn from the market. In other words, all these drugs have gone through 15 years of research, but still, once they were launched, after 6 months or 4 months or a year, drugs were withdrawn from the market because of the reasons. I'll talk about it. So you see, all these drugs, they are successfully launched in market, but in this many years, in 2013, 11, 10, 8, every year you will see few drugs will get withdrawn from the market. Why? Because you see serious cutaneous reaction, the side effect, lack of efficacy, no improvement in clinical benefit, which is lack of efficacy, increased risk of heart attack, hepatotoxicity, liver damage, risk of heart attack, liver toxicity. So you see hepatotoxicity. So these are the major reasons for which a drug has to be withdrawn from the market even after successful launch into the market. And the reason is, see, we try to mitigate all this risk very early in our clinical trials or in preclinical trials, but somehow when actually drug is launched across globe in different countries, we see that there are cases of side effects. When those cases are very minimal, it's fine. But if there are severe side effects in larger population, many cases, then it has to be withdrawn from the market. And as a science student, you unfortunately in our country, still few drugs are being used which are banned from the market. But having said that, government also in on DCGA site also you can find, I think they also publish this from time to time, the drugs which are withdrawn from the market. So that is for the general people to for their awareness. But as a science student, I just wanted to emphasize on that. Even after so much of research and effort, drug if it is launched into the market, there are reasons for which it needs to be withdrawn from the market because of lack of efficacy or safety or toxicology issues. Okay. So to summarize whatever we have discussed today, just that we start with hundred thousands of molecules in early drug discovery. We get into preclinical research where we do all in vitro, in vivo experiments. We do an IND submission. We get approval for clinical trials. Then we do phase 1 in 20 to 100 patients. Then we do phase 2 up to 500 patients, and then we do phase 3, and then finally we apply for approval, and once the drug gets approved, it is launched into the market. Okay, so would like to stop here, and tomorrow when we start, I'll start on this slide, but this is again, we will get into detail of that once you start with, say, target selection, you come all the way to a manufacturable target. From many molecules, you would have come to only one molecule at the end of it. So this slide in detail we will cover from in our tomorrow's discussion. Okay, so just to re-emphasize, you start with many molecules, but it is always that you end up with only one or two successful molecules into the market as a drug candidate. Okay, so this slide also will cover tomorrow, that yes, we have to have an assay in place for in vitro enzyme cell-based assays, or we do an in vivo study, then we do lead optimization, lead compound proof of concept, lead optimization, and then finally we want to do a safety assessment to have a successful preclinical candidate. Okay, with that I would stop here, and will be more than happy if there are any questions from the audience.
Yeah, thank you, sir. Thank you. Valuable information from your side about drug information, drug development, sorry, drug discovery and development. I'm extremely sorry about that.
In fact, we will be connected, Maheshwari, for the benefit of the students, that for next 2 weeks or so we'll be connected, so we can take more questions also, and I'll be sharing my email ID also. Students can post their queries, and I'll try and see that I can answer those questions. But in nutshell, the idea here is that we should really understand the nuances of the drug discovery and development process, and the students should really understand what all gets into drug discovery and development. Okay, and there is scope for everyone who is from life science sector in drug discovery and development. So at the end of the 2 weeks, I'm sure that be it a pharmacy student or a M.Pharm or a PhD or a B.Pharm or a PharmD student, or biotechnology, science, chemistry student, or a microbiology student, they'll be able to see where their role lies, okay, in drug discovery development, and where they can relate themselves. So if the purpose is that at the end of 2 weeks of this program, training program, if they are if they want to do their dissertation work, or if they want to pursue their research, at least they'll get some idea that yes, this is the area I would like to work further on, so that later on it will be easy for them to utilize that in their career. Okay,
Sir, one query.
please. Okay, so one of the question is there no stability examinations for drug discovery? The answer is
Sir,
yes.
your voice is not visible.
My voice is not visible. Hello.
Yeah, now it is
Yeah.
okay. Is there are no stability examinations for drug discovery?
Yeah. So the answer is yes. Drug stability studies are one of the important studies. So it really comes as is when you talk of drug discovery and development. So yes, it is a part of drug development. Like without stability studies, it is difficult to proceed further with having an approval for a drug. So of course, not in early drug discovery, but once your drug is clearing preclinical stage, when we get into clinical trials, simultaneously drug stability study efforts also goes on. In fact, in preclinical also, we start doing stability studies because that's very important. Because going forward, when you do when manufacturing, you should have the stability data in place. So yes, that gets into the place.
Thank you, sir, for clarification. Students, anyone having any query, please respond. I have given the feedback form in Airmeet, YouTube, and Facebook also. Please check it and give the feedback form. If anyone having queries, please put in a chat box in YouTube or Facebook. I will ask the sir.
Sure, definitely. And then let me. Okay, I will share my
The
email ID.
students are asking PPT. Can you share the PPT for
Yes,
students?
I will be sharing actually. By the end of this program, I'm going to prepare separate presentations for each of the topics, and I will share it. Of course, this is for the learning purpose, and I would want that as much students can benefit from that. So definitely, we'll be sharing the presentations.
Okay, sir. Students, as per sir instructions, we will share the PPT at the end of the day. Whole topic on drug discovery
Yeah.
development.
So before we end the course, this 15 days of course, we will share the topic-wise presentations, which you can utilize for your future reference purposes also.
Very nice explanation, sir. From your side, we learned so much today. From your side, really hats off for helping us.
Thank you,
Student
Maheshwari.
has
Thank you,
given
Maheshwari, and
student has given the feedback in the YouTube.
sure.
Yeah. Students, anyone having any queries, please respond. Otherwise, we'll close the section. And the feedback form is given to you. Based on that only, I will provide the certificates.
And for the students' sake, Maheshwari, let me explain how we're going to go about. So
Yeah, okay,
this
sir.
2 weeks of program actually we started from today with introduction to drug discovery and development, and tomorrow onwards we will deep dive into each of the topics, so that the whole course content can be covered. And what we will do is, uh, I understand all of you are really busy, and after this you will be attending your own lectures, or you'll be doing your research work and all. So daily we will revise, very briefly we'll revise what we have learned day before. So in nutshell, tomorrow once we start, we'll go through very quickly with the slides of today, and then we cover our new topic, so that what happens is that revision will be there for you. I don't want you to spend much time after this lecture learning these techniques, but of course, out of your interest, if you want to read well, but I will make sure that daily we do the revision of what we already studied a day prior, and then we take up the new topic, so that it will keep refreshing in your mind. And at the end of 15 days, on 15th day, we will do an assessment also. Okay, and of course, don't worry, it will be a very simple multiple choice assessment done for you guys. At least we will make sure that all of you can clear that assessment. Okay, so that is how we are going to cover this. And as I said, we'll be sharing my email ID also later on, and in the feedback form you can share the questions you may have, and I will try to answer those questions in the subsequent class. Okay, with that, I think if over to you, Maheshwari. I'm done. Thank you.
Yeah, thank you, sir. Sir, could you explain more about the programs by LSSDC? Student is
Yes,
asking.
about the apprentice programs of LSSDC. That's a good point, valid point. See, point here is apprenticeship. Actually, it's not a program from Life and Sector Skill Council, but what we do is we share once we, if you're interested, then you have to enroll for that, and then we send it across to the industries who are interested to take apprentices. And of course, we are running this courses also, online courses, but even otherwise also, since you are a graduate or a postgraduate, you are liable to get the apprenticeship opportunity. So we simply align you to the relevant industry, and if they have a requirement of your qualification and or your field, then they offer you the apprenticeship, and then they take you as an apprentices. So sometimes they ask for industry ready candidates. So that time, from License Council, we are running some courses that we can really help you. Actually, we can enroll you for those courses also, and then we can align you to the industries. But otherwise, directly after your graduation or a post graduation, you can be absorbed by the industry. We can really facilitate that action. I mean to say, we can make sure and we can align you to the relevant industry for your apprenticeship opportunity.
Okay, sir. Thank you. Students, anyone having any queries, please respond. And I have given the feedback link to you in YouTube and Facebook also. Please check it and give the feedback form. Based on that only, we'll provide the certificates. Anyone having any queries, please respond. Students, anyone having any queries, please respond. I have given the feedback form to you in YouTube and Facebook also, and Airmeet also. Please check it and give that feedback. Based on that only, we'll provide the certificates
Yeah.
taking the academic.
Meanwhile,
And anyone having,
yeah,
yes, sir.
meanwhile, somebody has just posted, is this computer-aided drug designing? Yes, we will be covering computer-aided drug designing topic also. So, if that's the question, then yes, we will be covering computer-aided drug designing also within the our course. Okay. Today was just the
Yes,
introduction
sir.
to drug discovery development concept. When we go phase-wise, we'll be covering computer-aided drug designing and development also.
Okay. What are the benefits that we are going get through the LSSDC?
There are a lot of benefit if you align yourself to LSSDC. I'll tell you, because Life Science Council is a non-profit, first of all. So its goal is not to get profit. It's an initiative from the government, and our only mission is skill development. And we do whatever needs to be done to encourage more and more skill development. Like one of the thing is, like, we did a collaboration with APSSDC because we want to participate in skill development. Like that, we have aligned industry with us. We have aligned academias so that we can provide and facilitate to fulfill the requirement of the industry by providing them industry ready candidates. So what do? We run Epartial, which is again an accelerated 1 week courses where after that training we really teach students in a crash course manner in just 1 week of time about the subjects. Like that, we are running Ecertification programs where it's a 1 month program where we run those machine operator and quality analytics and all those things over a 1 month e-learning program. And apart from that, as an apprentices, as I said, we help you align yourself to the relevant industry in your area so that you can be absorbed as an apprentices as and when industry has a requirement. Okay. And even in future, whenever we come up with we conduct webinars on a regular fashion, purely for they're all free of cost, purely for the education purpose, where we cover different programs of LSSDC, and we take any technical topic and we go in detail of that.
Yeah, thank you, sir. Thank you so much. No one having any queries again.
No worries.
Thank
On
you.
a lighter note, Maheshwari, I would like to say this. I always believe in my trainings that there are only two scenarios. One is either you have understood everything, then you may not have any questions. Second is you have not understood anything, then also you won't have any questions. So I would believe that the first instance is true, and all the students have understood everything. That's why they don't have any questions. So
Mhm.
thank you all of you for your time and really sparing your precious 2 hours, close to 2 hours. Watching this video or participating in this training program, and I'll try my level best to make it as interesting as possible. And in next 2 weeks of time, I'll take you through this entire journey of drug discovery and development. And at the end of it, I would want that all of you should have an overview understanding of drug discovery and development. And if any way I can be of any help to you in your dissertation work or your research work or your industrial training, summer trainings, I'll be more than happy to do that. And once again, my heartiest congratulations to APSSDC for this wonderful initiative of skill development via e-learning, and looking forward to this program of next 2 weeks or 15 days of program. And I'm sure many more such programs will be coming up in near future. And once again, good luck to APSSDC, and looking forward to a very live, interactive, and a very useful session in coming days. Thank you. Over to you, Maheshwari. Thank you.
Thank you, sir. Thank you so much for blessings will to us. And today our attendance is 1495 students have attended, sir. From the starting to ending, they are alive.
Oh, good to know. Thank you, ma'am. It's really, highly,
Thank you, sir.
it is very motivating. Thank you.
Thank you, sir. Thank you so much. Bye bye, sir.
Bye. Thank you.
స్టూడెంట్ ఫీడ్బ్యాక్ ఫామ్ అందరికి యూట్యూబ్ ఫేస్బుక్ లింక్స్ లో ఇచ్చానమ్మా, చెక్ చేసుకోండి. చెక్ చేసి ఫీడ్బ్యాక్ ఫామ్ ఇచ్చానమ్మా ప్లీజ్. I have given the feedback form. Please check it and submit the feedback form. Based on that only I will provide the certificates. Okay. Thank you everyone. The section is closed. Bye bye.